Good morning. My name is Rachel Gjerde. This is my proteomics paper that I'm going to share with you today. Let me just get my screen going. Okay, so my chosen proteomics paper is dealing with SILAC, which is stable isotope labeling by amino acids in cell culture. This is the title of the paper that I chose. It's called Profiling the Skeletal Muscle Proteome in Patients on Atypical Antipsychotics and Mood Stabilizers. The last slide in this slide deck also gives the reference for the paper. So that's the one there that you see, and the authors are listed here. Okay. The objective of the study, they're trying to determine if there are differences in the amount of the proteins in the skeletal muscles between patients on AAPs, which are atypical antipsychotics, and patients on mood stabilizers. And the results do indicate that indeed this is the case. There is a difference in the amounts of proteins in these two types of patients. AAPs are used to treat mental illness. In the case of this study, they're talking about people that have bipolar 1 and bipolar 2. One of the problems with people on these medications is that there are a lot of side effects. So people tend to not want to take the medication. So if they're prescribed the medication, they don't take the medication. Weight gain is one of the side effects. One of the main side effects is insulin resistance, and that means that your body can't use insulin effectively to absorb glucose, and your cells need that glucose for energy, so to do everything that they need to do. So there have been other studies on the effect of these AAPs on the human proteome, and those studies looked at blood serum or blood plasma, but this study claims that using skeletal muscle would be a better fit because that's where glucose uptake actually happens within the skeletal muscle. So they're trying to study this insulin-stimulated glucose uptake and its deregulation in the skeletal muscle itself. There have been previous studies that did also use skeletal muscle, but that was in rats, not humans. So this is using human skeletal muscle. The background information in the introduction section here was definitely adequate to understand what the aim of the study was. Let's see, did I cover everything there? Yes, I think so. Okay. So let's look at the methods. Okay, so first thing we're going to talk about are the sample types. So the patients here have been diagnosed with bipolar 1 or bipolar 2. There were 16 patients chosen for the study. So each patient had that diagnosis. There were 8 patients in each cohort. So 8 of the patients were taking an AAP, and the other 8 were taking some sort of a mood stabilizer. So from those 16 patients, they took muscle biopsies, and then they froze those muscle biopsy samples under liquid nitrogen. So that's where they were getting the samples from. In the next step, they took the samples and they put them into a buffer that contained urea and protease inhibitors and phosphatase inhibitors. So this step was meant to break open the cells, so lyse the cells, solubilize the proteins, so basically dissolve the proteins in that buffer. And then also it preserves the protein's native state. So in other words, it preserves what state the protein is in, and apparently phosphorylation is important here. So it saves the phosphorylation status of the protein in its native state. The urea is used to denature the protein, so it breaks them apart, making them more accessible for the analysis, and also prevents them from aggregating or sticking together. The protease and phosphatase inhibitors help the sample not to degrade and helps the sample the proteins don't get modified further by any enzymes that are already present in that sample. So they've got their samples, they're broken apart, and then they're going to spike those samples with a small amount of heavy SILAC labeled protein standard. So that's that, they call it universal SILAC. So that's the stable isotope labeling. So These authors claim to have developed this method. It's called universal SILAC in a couple of their previous experiments. So basically what happens is they label or they add this small amount of this SILAC standard, and then they can compare their sample to the universal standard to figure out the quantity of each of the proteins in the sample. And then once they've got their SILAC labeled protein standards mixed with their actual sample, they're going to use those samples and they're going to digest them and then analyze them using HPLC, ESI, MSMS to check on protein abundance, and that we'll talk about that on the next slide. Okay, so what does that stand for? High performance liquid chromatography, electrospray ionization, tandem mass spectrometry. That's what we're talking about here. So what they did first with their samples is they separated them using the HPLC. They use what's called a C18 reverse phase column that was packed with something called Reprosil Pure C18AQ3 micrometer resin in a buffer solution. And then they passed a 5 to 35% gradient of another buffer through that solution. So the instrument was used to detect the abundance of the proteins, and that was called a Nanoflex Dionex Ultimate 3000 UPLC system. And it was also coupled to a Thermo Finnegan LTQ Orbitrap Lumos, which had a Nanospray Flex ion source attached to it. So a little bit about the system here. So the Dionex Ultimate 3000 was the HPLC system used, and it's said to be ideal for small sample volumes, which is what we're dealing with here. And it's something that's often used in proteomic studies. The LBQ Orbitrap Lumos, that's the mass spectrometer that was used. And it combines 3 different types of mass analyzers: a quadrupole mass filter, a linear ion trap, and then an Orbitrap mass analyzer. And again, this is a system that's often used to characterize proteins, especially when they're in low abundance. So in order to get the proteins into the ion form, they had what was called a Nanospray Flex ion source. And what it does is it applies a high voltage to the liquid samples, and this produces charged droplets that are then analyzed by the mass spec. Let's keep talking about the methods a little bit more. So they used what was called a top 20 approach to identify their peptides. So what this means is that a full scan of the spectrum of that sample is done, and then the top 20 most abundant ions are chosen from that full scan. Then those specific 20 ions are subjected to CID, which is collision-induced dissociation, so breaking them apart further. And then the mass spectra of each of those broken apart ions is obtained. So the Orbitrap mass analyzer completes the first scan, that full scan, and then the 20 most abundant peaks are fragmented with the linear ion trap. So then they took that information and they had something called the Max Quant software, and it helps to identify and quantify the proteins. So that's the first thing that they did. Then there was a section in the paper about the statistical analysis that they used to determine which types of proteins and how much. From my limited understanding of that, it seemed to be appropriate. The first thing they did was they used some filters. So they were trying to narrow the number of proteins that they're going to compare between these 2 groups of patients. So one of the first filters they used, they said that each protein needed to be identified in 4 or more biopsies out of the 8 in each group. And then the second filter they used was if there was a change in the amount of protein, it needed to be at least a 1.5-fold increase or a 1.5-fold decrease to be included in the results. So those were the 2 filters that they used. The study also used something called in silico modeling. Basically, it's using computer simulations to determine something called the pathway analysis. So this means they compared the identified proteins of interest that they got from those top 20, and after filtering, and then they would compare that with a reference database of known pathways to determine whether the abundance of a particular pathway is more than what would be expected by chance. And the software they used to do this was something called the Ingenuity Pathway Analysis software. So basically, they're trying to figure out, does this pathway of this protein increase more than you would think it would. Pathway analysis, and we'll talk a little bit more about that later. Okay, so now we're talking about some of the results. So again, as we stated earlier, there were 16 total patients, all have bipolar disorder. 8 are being treated with AAPs. The other 8 are being treated with some other type of mood stabilizer that is not an AAP. So basically, 2 different types of medication is what we're talking about here. They did say here in the results section that some of the patients are diagnosed with bipolar 1, 13 of the patients, and some are diagnosed with bipolar 2, 3 of the patients. And it was a mix between which group those patients were in. Bipolar 1 and bipolar 2, the main difference between the 2 diagnoses is that it has to do with how severe their manic episodes are. This might have impacted the results. They didn't really discuss that within the paper. They brought it up, but they didn't discuss how they thought it could have maybe impacted the results. Also, the patients were not all treated with the same AAP. So there are different types of these atypical antipsychotics. So in the results section, there's 4 different AAPs that are mentioned that different patients are being treated with. And also 3 different mood stabilizers are also mentioned. So again, this might have been something that could have impacted the results. And it was brought up in the paper, but not really discussed how do they think that could have impacted the results. So within the samples, there were 1,110 proteins identified within those 2 groups of patients. Again, remember, they filtered out those proteins. So again, they decreased the amount through filtering. So each protein needed to be identified in 4 or more biopsies out of the 8. And also any change seen needed to be either a 1.5-fold increase or a 1.5-fold decrease. So they went from those 1,110 proteins and filtered it down to 168 proteins. So then they took those 168 proteins and they did a statistical t-test on each protein to see if there was really a statistical difference between the 2 groups for each of those 168 proteins. Of those 168, 34 were determined to be statistically different between the 2 groups. So basically, they've taken the 1,110 proteins and filtered it down to 34. The second thing that they did, because they're saying a total of 40 proteins had a significant difference. So where did those other 6 come from? So the other 6, they said if a protein was identified in more than half of the samples of one group and not identified at all in the other group, they assumed that it was too low to be detected in the second group. So they added all proteins that had that filter, basically, or that definition to their original 34, and there were 6 proteins that fit that definition. That I thought that was a little bit questionable, making that assumption. So now we've got 34 plus the other 6. If they're only identified in one group, again, they're assuming that it was just too low to detect in the other group. So now we're up to 40 proteins that have a significant difference in abundance between those 2 groups of patients, the AAP patients and the mood stabilizer patients. The next 2 slides are going to give you a list of those proteins. So I'm just going to quick look at those. So here's, so we've got the protein name, which gene it's found on, and then the two cohorts, the AAP, so the atypical antipsychotics group, and then the mood stabilizers group, and then something called a p-value, which was how they decided if this was what's the word? Statistically significant, was using that p-value. So here's the first half, and here's the second half of those proteins. So those are the 40 proteins that they've identified. Okay. There is also, on the next slide, their pathway analysis results. So they identified 107 total pathways showing enrichment. And then after the statistical analysis, only 21 of those pathways were seen as statistically significant. So this is a list of the pathways. I know it's kind of hard to see. Sorry about that. It's a little bit blurry. The top one, though, was what they thought was one of the most significant. So it's the P70S6K signaling, and I don't know if I'm saying that right. And then on the right-hand side, you can see which proteins were assigned to each pathway. The experiments done do seem appropriate based on the objective of the study, and the results make sense, although I do question their inclusion of those 6 proteins based on an assumption and not really a specific measurement. I've left out some of the figures that were in the paper. I don't really felt like they added much to the study itself. One is a table of the data on the 16 patients, just saying like what their blood pressure was, what their BMI was, just some information about the patients in each of the 2 cohorts. And the second is a set of pie charts that shows the location and the functions for the 40 proteins identified based on if they were measured as increasing or decreasing. The legend for those pie charts is pretty vague, and it doesn't really bring a lot to the table. It doesn't seem very informative to me. It does seem like they did appropriate statistical analysis on the data in order to interpret the results, because they talked quite a bit about the statistical analysis. So we're on to the discussion. So why is this study significant? Basically, it's the first study that compares the proteome of the skeletal muscles of those AAP patients to those that are on mood stabilizers, so not AAP. Again, previous studies used blood samples, and this is the first time we've got skeletal muscle samples. They believe that the side effects of the AAPs are most likely occurring because of the changes happening inside the skeletal muscle. So that is the importance of using the skeletal muscle. Future studies they've talked about could look at the overlap in the findings between the blood sample studies and the skeletal muscle studies. One of the most significant pathways that I talked about in the previous slide is the one that's listed here, the P70S6K signaling. And this might be involved in insulin sensitivity, which again is a concern for patients taking these medications. So they're seeing what they were expecting to see, that there would be something to do with insulin sensitivity. There is also significant enrichment in several other pathways, more than could be thought of as normal, including those for glucose metabolism, cell cycle, apoptosis, which is programmed cell death, eventually cells die, and folate metabolism. The folate metabolism one is talking about folate-dependent one-carbon metabolism, and that's something that's critical for cell growth and survival. It produces NADPH and nucleotide precursors, and that occurs in both the cytoplasm and the mitochondria. In this part of the paper, the discussion section, there was a lot of discussion about several different pathways that might be important, and signaling pathways that might be important. The paper did note here also that the sample size could have a limitation on the results, which is something that I thought of. There's only 16 actual patients here. And also they did discuss that possibly those different medications taken by the patients could be important. So again, we've got some on AAPs, but not the same AAP. And we've got some on mood stabilizers, but not the same mood stabilizers. And again, they did talk about some statistical analysis that was done to help decrease the number of false positives, but they did note that there could possibly still be some false positives. Several of the specific differences were also discussed in this section of the paper, but I'm not going to go into those here. And in addition, some changes were discussed in terms of where the proteins are located, what their function is, but again, that was very vague in that section of the paper, so it didn't really add much to the discussion. Let's see. Some final thoughts. I do question the fact that such a small sample size was used for this experiment, but based on the amount of work it would take to work through one patient's data, I do understand why they started there. They wanted to see if there was some sort of a difference that they could see, and maybe in the future they'll increase the sample size. In order to determine if this work is biologically significant, definitely need more patient data that should be included. And also, I think it would be best if all patients have the same diagnosis, either bipolar 1 or bipolar 2, because again, that might make a difference. And if all patients in each of those groups, the AAP group and the mood stabilizer group, I think it would be better to have each on the same medication, because again, that could be something that could make a difference. Really, the only one of the variables that was not discussed by the paper's authors was that difference in diagnoses, the bipolar 1 versus bipolar 2. And I don't know enough about bipolar 1 and bipolar 2 to know if that is important or not. Thank you so much for listening to my paper, and have a great day.